Good morning, everyone. My name is Pedro Souza, and I'm a professor at the Faculty of Dentistry at the Federal University of Goiás. Thank you all for attending to this event today. It's a pleasure to be here today for the first international meeting on dental research organized by our faculty. First of all, I would like to thank Professor Lídia Jordão and Túlio Nogueira and all the organizing committee for the support that made possible for this session to happen. In this session, we will be talking about entrepreneurship in the university, from the idea to the application in the development of biomaterials. I have two very special guests, Dr. Gian Nutal from ETH Zürich and Professor Morten Foss from Aarhus University, Denmark. From now on, I will just call them Gian and Morten, and I will ask them to please call me Pedro because it's easier to talk to friends using the first name and we don't need to worry about that. The aim of this discussion today is to talk how entrepreneurship is encouraged in the different countries, how are the opportunities, what's needed to be an entrepreneur, the experience of Morten and Gian and on entrepreneurship and so on. Morten and Gian don't know each other, they just knew each other today, but I'm sure that there's a lot in common and we will find out that today. We will start the presentation from Morten. He's a senior researcher at the International Nanoscience Center, iNANO, at Aarhus University. I'm collaborating with him since 2018. And from there, like, we have some students interchange, and students from Denmark came here to Brazil. Students from Brazil went to Denmark. It's a very nice collaboration. And having a PhD in physics, he's the leader of the biomedical surface group. He's also the Danish principal coordinator for the nanoscience research area at the Sino-Danish Center. And besides all that, he also likes nature and sports, and he has successfully climbed the Morro dos Pirineus in Pirenópolis when he came here to Goiânia. Morten has some patents, including one that is strontium coating for implant devices, that is highly applied to dentistry. And the main objective of his research is to gain insights into the interaction between artificial and biological systems for biomedical relevance. So Morten, thank you very much for accepting our invitation to be here today. And the floor is all yours. Please start when you feel like starting.
Great. Pedro, thank you for the very nice introduction. I would also start by thanking the organizing committee for setting up this, and Pedro, thank you for inviting me. So I guess what I will do today is I will start a little broader, as you said, Pedro, about so what do we do where I'm from to help the students getting this entrepreneurial mindset, and then I will go for a few examples that we have been through all these stages in my group. So it all started, as you can see here, in 2018, where Pedro and we got a network program grant that with the name Biomaterials Meet Bone Biology. And here you can see, in fact, Pedro and his group visiting iNANO Center at Aarhus University. So I'm from Aarhus University, and for the people who do not know where Denmark is, it's just below Norway and Sweden and just north of Germany. And I'm from Aarhus, which you can see here. That's the second biggest city in Denmark. Copenhagen is somewhere over here. And we have as you can see, a very nice campus. We have approximately 50,000 students and 11,000 employees, 2,000 PhD students. So I would say probably in Denmark, it's a big university, but you would probably consider it a mid-sized, maybe even smaller university. So I'm part of what is called the iNANO Center. So that means that's an abbreviation for the Interdisciplinary Nanoscience Center. So it's focused on nano, but what is also important is that we try to bring together people with different backgrounds, as you can see here, physics, chemistry, biology, molecular biology, IT, math, and then we also bridge all the way to health, to the Faculty of Health. So the idea is to bring people together and get new ideas and new also bringing products, in fact, all the way to the market. So we have a threefold mission. Of course, we have to do good research. So the idea is to strengthen this interdisciplinary research in nanoscience and also catalyze new collaborations with other international research groups, for example, the group of Pedro. Then we have to educate people. That's one of the important things in any country. So we want to play a key role in the education on different levels, the bachelor level, the master's and the PhD, and all the way to the postdoctoral levels. But then the third one, that's this one, the innovation. And that's what I'm going to talk about today. So what we really aim for is to provide a good interface for transferring our basic research science up here into, in principle, all industry, preferably Danish industry, but also bringing it all the way to society. So the question is, of course, how do you create such a mindset for the students? So we have a bigger setup at the university where we have different people helping the students when they get that far with an idea. That's what they call the tech trans office. But before that, we are also trying to give the students this mindset of an entrepreneur. And in fact, what we have at the Nano Education is a compulsory entrepreneurship course. And I'm part of that as well. And basically what we're trying here is we're trying to teach the students, first of all, on a more science-based level, what is entrepreneurship. So they are getting articles about entrepreneurship, learning about different methods. How can you approach problems or challenges found around in the world? And then in fact, we let these students learn through the whole process. So we are letting them go through all the process from identifying a problem, trying to find solutions, validating these solutions by contacting different opinion leaders, and then going all the way to a pitch and in fact also a business model. And so the idea here is to give them what I would call a near entrepreneurial experience, but still, you could say, in the safe environment of the university. So you don't risk your house, you don't risk all your money, but you still have try to give them this experience of how is it to go from an idea or identifying a problem all the way to hopefully raising funding and getting a product on the market, including prototyping. So this in general has been pretty challenging for the students. And each year we are normally giving them a specific, what we call an arena. Instead of just saying, find out a problem in society, we typically set a stage that says, this year you should look into this area, but it has to be broad enough that everybody can bring in their expertise. So for example, I was back looking what we have been doing earlier. And for example, one year that I think that was like 4 or 5 years ago, then in general, the arena was health in the aging population. So as you can also see, this is very broad, right? Because as the definition from WHO, it could be health is both physical, it's mental, it's society. So there would be a lot of things you could try to identify within this area, right from a biomaterial, from a new implant to an app where you bring people together socially. So this is a challenge they would get, and then they would have to identify a specific problem. So we, of course, which I think is natural at a university, some students would say, and they do say, why do we have to do this? Why do we have to learn? Because in principle, we go to the university because we are academics. So that's what we want. We don't want this. And then at least what we're trying also to teach the students is that you could say in many ways your research and such a startup innovation process are two sides of a coin. And why that? That's because you have many of the same challenges. You get new problems that has never been solved before. You have to keep on innovating. You should never accept status quo, and you have to push the problems using your creativity, and also having this mindset about things might not work, but then you have at least shown that this does not work, and you have to find a new way. And you normally people are very good at being trained to think, how can we solve this? Why did this happen? So in principle, I see many of the same skills. You can sit in the ivory tower, but you are still a kind of an entrepreneur. So I think that's important to convey to the students, that you are thinking in an innovative and entrepreneurial way, even if you're only, so to say, only doing research. In fact, also if you look in general at the CEO level internationally, you would also see that there are many, for example, chemical engineers that sit on top positions around the world. And why is that? This is exactly because this is what they have been trained for. Maybe not specifically what the company delivers, but they have been trained for a way of thinking. So that's at least what we're trying to tell the students. And I would say, if I look at where our students go, so this is, I guess, some of them are Danish companies. You would not know them, but I can tell you that our students in general go to different kinds of industry, also academia, but definitely also industry. I get, McKinsey, for example, here, right? Some Vitrolife, so some EPFL. So our students cover right from consultancy to hardcore physics to, again, Novo Nordisk, as you would probably also know. So I think in that way, we're trying to teach the students to go out and have like all these possibilities afterwards. I could give you many examples of this, but of course, what I would prefer is to give an example coming from my own group. So this is what I will do. So I have this biomedical surface group at iNANO, and in principle, what we are looking at is everything that involves an artificial material and a biological system in general. And basically, we have focused on four areas. We have the bone contacting implants that many of you for sure know very well. You can see here, for example, an artificial root. We develop non-fouling surfaces, for example, for catheters, or we go for percutaneous implants. That's implants that penetrate the skin. You could say that also has applications in dentistry. And then we are also looking at what is called large-scale screening of cellular response. That is there a way to screen a parameter in a systematic way, and in that finding out how the cells react to these cues on the actual surface. So one example, So my background is physics, and that is why, in principle, I like to bring something all the way to the clinic, but I also like to vary one parameter at a time, because sometimes it makes you easier to get these guidelines. So what I will do is I will start with this one, like some soluble factors and how this can control cellular response. So this was a project that was started, I guess, many years ago, more than 10 years ago. And we were looking at osseointegrating implants. And one of the things that when we talked to like industry and surgeons was that one of the things that could, in general, osseointegrating implants work, but what they really would like was a decreased healing period. And they had a problem with micromotions in general. And also they said that if a patient has a less good bone quality, for example, osteoporosis, then sometimes the implants still do not work. So that being said, we thought about how could we help integrating such an implant. And one thing we knew was that at that point in time, osteoporosis was treated with something that was called strontium granulate. So that's you can see here an organic compound where you have strontium ions complexed to it. And it was also known that there were some side effects on the strontium treatment. This was given orally typically. So there was some side effects that were pretty severe, for example, DVT. But the question was then, so that was a systemic thing. Is it then possible, in fact, to do a local delivery of strontium right around the actual implant? And by the way, I'm sure many of you know strontium, at least from New Year's Eve. If you see fireworks, then the red color is an optical emission at the actual strontium. So the idea was to try to develop something where you could deliver the strontium that we knew would work on bone regeneration, but without the side effects. So of course, a sustained release from the actual implant was an obvious way of going. And it was also, we also knew from literature, there's a lot of literature out there that the strontium, what it did was it increases the effect of the osteoblasts or the activity. So that means it increases the bone buildup, but also it inhibits the osteoclastic action. So that means less bone resorption, more bone generation. So that was fine. So what we decided to do, again, also because my background is physics, was to try to do what is called a PVD coating, physical vapor deposition. So the whole idea here is that you have some targets, as you can see up here, and this you can change the composition, the chemical composition of these targets. And what we did was we combined strontium and titanium in these ones. Then what you can do is you can remove atoms from the two actual targets here, and then deposit these atoms on a substrate, which in this case could be implants. So we did that, and as you can see here, this is a cross-section SEM image, sorry. And you can see this is around 1 micron coating. This is the substrate down here. So this coating consists of titanium and strontium, and then there's some oxygen in it as well. So what we had to do now was we had to try to, first of all, we had to make sure that strontium was actually in the coating, and we did this by something called XPS, X-ray photoelectron spectroscopy, which checks the chemistry of the outer few nanometers of a surface. And what we could see for sure was that we could see the strontium. I mean, don't mind the actual different what do you call it, like the oxygen 1s and so on. But what is important is you can see the SR3P, so there was definitely strontium in the coating. We also knew that we had to deliver it, so we did washout experiments in PBS, washed out on these samples with the strontium coating. And then what it did was something called ICP-AES. It sounds complicated, but what it is simply a technique where you burn off some liquid and then you check for atomic emission. That means, again, looking at optical different photons. And then what we could see was we found the red color. And so we could quantify the amount of strontium, and it was released as we really wanted to. So we knew now we could release the strontium. So now what we did was we optimized the different coatings to optimize the actual strontium release. And we found out many things that maybe is interesting for a physicist like me, maybe not that interesting for people that just want to apply it, but at least there was not a simple correlation between strontium and the release, which I think was interesting for me. But we could optimize this, and the first thing, or one thing we did, was to go for in vitro experiments, and we used something called periodontal ligament cells. And the next question would then be how, if we want to add strontium to the actual medium for the cells, right? Which concentration should we use? And that was not a simple question, because the question is, if you put in an implant in bone, how big a volume is this strontium in fact leached to? So we tried to estimate it by a healing chamber model, and we estimated that maybe like a quarter to half a millimeter, that was the volume where the strontium was released. We knew how much was released, so then we could calculate the actual concentrations. We did some in vitro, and basically what we found was that for high strontium concentrations, as you can see here, for example, this is PCR gene expression, this is osteopontin, osteocalcin. As you can see, for the higher concentrations, there was a significant effect on the gene expression. So that means that for sure the strontium did have an effect on osteopontin, osteocalcin, and you could also see this is proliferation, days, number of cells. You could in fact also see that for the higher strontium concentrations, we also had a higher proliferation. So that was positive. But of course, in vitro is good, but you have to go to in vivo. So the next thing we did was we went to a rat study. I guess this, you can see this is a rat, a Wistar rat. And if you zoom in on these red circles, what you can see is in fact implants here. So what we did was we had a press fit implant that was coated with this strontium coating, kept it for 4 weeks, sacrificed the animals, and had a look at the did histology. And as you can see here, this is the femoral shaft, a cross section. This is the control, this is the coating, and this is 2 images put together. And this is a bone marrow, the cortical bone around here. And for sure, I guess you can see that there's much more gray over here than over here. So it definitely seemed like this coating had in fact worked. One thing that worried us a little was that we didn't see any bone here. And as you all know, titanium is a really good biomaterial. So we had this hypothesis that this was most likely due to small motions. So that means maybe if we had a screw instead of just a pin, that might improve the results. So, but also when we had these results, what happened was that, as you can see down here, we filed a patent, as Pedro mentioned, and that patent, I can tell you now, is now licensed to a Danish company. So we optimized further the coatings, and this is very physics-like, so I think I'll drop that. For me, it's interesting, maybe not that much for you, but you can change the parameters of coatings by changing background pressures and so on, and rates of deposition. And in that way, you can also optimize porosities, and in that way also optimizing the actual release. So this what we used a lot of time on optimizing this, also published. And then we designed a new implant again. Now it has a thread, and that should then a thread, and that should then maybe stop this micromotions and maybe improve the results. So we did another in vivo experiment, and what we saw here, again, we have the femoral shaft here, but what you see now, again, this is the good coating, the strontium coating. This is the grade 4 titanium, and now what you see is a lot of bone over here, and also what we had hoped for, also a nice thin layer of bone on the, you could say, the control grade 4. Very positive. So we were at that point, we were kind of really happy about the development. So it worked. We published, and as you can see, we published 3 articles. So that was, you could say in principle, as a scientist, that was it. We have proven that it works. But of course, if you want to push things all the way to the bedside, maybe being part of producing something that is a real product, then what you have to do is not just to show that it's better than titanium, but you have to show that it's better than the best on the market. So The next thing we did was we bought different implants that we knew had a really good performance, and then did another group with our titanium strontium oxide and made a rapid study, because that would then prove if we could match what is actually on the market. So looking at histology, then the ROI 1 and 2 means very close to the implant, up to 250 micron from the implant surface. ROI 2 is from 250 micron to half a millimeter. And as you can see here, SR is the actual coating, and you can in fact see that it's definitely just as good, but most likely it's not just most likely, especially a little away from the implant, it's in fact much better than the other groups. So that was really, really positive. And you could say this is right now the end of the story, because it's right now being evaluated by that company for an actual product. But one thing we have done after this is this about understanding where does the strontium actually go. I think that's of basic interest. We had this hypothesis that it would be in this healing chamber up to 500 micron away from the implant. So what we did on some of these histological sections, we did synchrotron radiation fluorescence study. So here what you do is you shoot through a thin sample, and then you simply again look at the fluorescence from different types of atoms. And as you can see up here, we have a coating, and this is control. And then the red here is calcium. Of course, there's calcium in bone. Green is the titanium, and we have the screw here, as you can see also here. And blue is now the strontium. And what is interesting is, I guess you can see that there's a very thin layer of strontium. That's the coating here. But what you can also see is that the bone is more purple here. So that means strontium has in fact leached out and is now incorporated in the bone. And what is really, really nice is that this length scale is 500 micron. So that means, as you can also see, that most of the strontium is in fact leached in the closest 500 micron to the actual implant, which again, in fact, validated our, you could say, our guess, best man's best guess at that point in time. So this again shows that it stays local. So this is one example from you could say our development, and I think what we could learn from this at least is that first of all it takes a long time as soon as we have anything involving medical devices. So we started, I would say, now it's more than 10, it's probably 15 years ago, and we have worked on this ever since. So now we have something that is close to a product, so it takes time. What is also important is to have the right student involved. I got a really good student that also wanted to pursue such a product and also had an interest in seeing this on the actual market. And as a side thing, I can tell you that what happened with that student, he did his PhD on this one, and this project, what happened is that as soon as he finished, he got a job in the industry, that in that company that in fact licensed the patents, because they could see the value in his background. Also, another thing that you have to think about from the start is that it's really good to have the right competence and the right companies involved right from the start. So I think what was nice with this project was that we got in contact with a company that could push this all the way to the market from day one, and they could then push us as academics to maybe be more focused on the market, while still, of course, having this in the back of your mind that you have to publish. So I think in that way, in many ways, this was a successful project. This was only one part. This was, you could say, soluble factors, sustained release, but we're looking at many other things in the group. We are looking at stiffnesses, shapes of structures, different topographies, and also biochemical factors at the actual surfaces. And I don't really have time for all this, but I would just give you a very brief example of another thing that we did, which was involving this materiomics, which is screening of structures. So basically here the question is, when we have all these parameters that can control cellular and tissue reaction, how do we optimize these physical parameters? Because we have a multidimensional space, so it's really complicated. So one way is a high throughput screening of these interactions. And we developed that many years ago. Here you can see the initial screening platform. This was 13 by 13 structures. So as you can see over here with the SEM images, so it's in fact a simple mountain landscape that is very well ordered. So what we did was we changed parameters in a systematic way. Then the next thing would be to seed a specific cell type on all these structures in one go, and then having a look at how these cells, they reacted. And the first cell type we used was MC3T3s, that's the murine preosteoblasts, and this is alizarin red staining. And as you can see here, then there's a lot of red here, less red down to here, a lot of red here, again down to here, a lot of red starts again here, and that fits with these dark colors, which fit to a specific periodicity. So that means we could, with this way, we could identify mineralization in vitro with a specific design, a specific periodicity of the structures and size of features, which was very nice. So we could, again, here what you see is these ones correspond to size of pillar and distance in between. So here what we have is we changed the distance between the pillars, increase the size of the pillars, change the distance again in between, and this is mineralization on the y-axis. And as you can see is that for small structures, we have a very high mineralization. It decreases when the distance between the structures gets too big, and the same then repeats with larger pillar diameters. So that was very intriguing for us. And what I found especially intriguing, again as maybe a physicist, mathematician, is that when we are talking about roughnesses, then if you calculate that mathematically, then a structure, if you double all distances on a surface, then the roughness is exactly the same, the standard roughness parameters. So for example, here a 1,1 that had a lot of mineralization, in principle has the same roughness as this one, the 2,2, the same as the 4,4 and the 6,6. So this again shows, I guess, what you all know, that roughness is not a really good predictor. It can help, but it's definitely not a good predictor for cellular response. But this was an easy way of seeing this. So I have used my time, but I would just use a few more minutes. Of course, we did a lot of experiments here. We checked for osteopontin, osteocalcin, alizarin red, and all that fitted very well. But what I definitely just wanted to say is that such a platform, we're still working on that. Now we are at the nanoscale, has, I think, a lot of possibilities to screen, because this was, you could say, osteogenic differentiation, but in principle, you could do many other things. And we also did that at this point in time, again, imagining that what we could do was we could produce smart petri dishes, identifying good structures, and then go to petri dishes and then guide cells in a specific direction. We have also looked at embryonic stem cells, for example. And in fact, what we did was we submitted 3 patents at that point in time. And whether that was a good idea or not, I don't know. But at least what happened was that a big international company acquired these 3 patents. So there was no spin-out company, but a sale, which in general is not what Aarhus University really wants, because they prefer, which I also think is in general a good idea, is to push or at least help the actual employees and the students to create a spin-out, a startup. But in this case, that would not happen. So I could continue a lot because we have done different cell types. We have done nanoscale topography is what we're doing now. I'm doing this together with Pedro. But I think in order to keep time, I've used a little more than half an hour. I think I would stop here and thank you all for your attention. And I guess questions is afterwards, right?
Yeah, thank you very much, Morten. It was a very nice presentation. There are some questions, and I would like to also to ask you, but we leave the questions for the end. And at the end, we can discuss and talk also to Gian, that we will talk now.
Yeah.
Thank you very much. It was very nice. Thank you again for accepting to participate in this session. And now we go for our next guest. Gian, I think it's the first time I say Gian, because now I learned I say Gian, and everybody will understand Gian. Holds a PhD in bone tissue engineering and imaging processing from ETH Zürich in Switzerland, and he's currently a postdoc at Professor Ralph Miller's group. I met Gian this year when I was a visit professor at ETH Zürich, and I went to visit Professor Miller's group. And in the first meeting, I saw Gian asking questions, and I already noticed that he was kind of a special guy, always asking very good questions and relevant for the presentations. Gian and then I invited him to go to a restaurant, and we had a very nice, in a pop-up Vietnamese restaurant. We had a dinner there, and now he's launching a startup called CONFAP that is based on the results from his PhD thesis. We discussed a lot his idea and his goals during my stay in Zurich, and I thought it would be very nice and interesting for you to listen to him today. So I invited him to participate. Thanks for accepting, Gian, and you can start whenever you feel comfortable starting. Thank you very much.
Thank you very much for the invitation, Pedro, to talk today. It's a pleasure to give this talk to so many people. So I completed my PhD this year. I defended in May 2021. And what I wanted to convey now during my talk is basically how came I up with an idea, or with this idea, and how did I transition from doing the PhD towards, okay, I want to launch now a startup, and then what plans do I have actually for the startup. So the startup, Peter already mentioned it, it's named CONFAP, and the slogan that we have at the moment, and it always changes a bit because we're still developing the business model. It's basically the first animal-free bioengineered bone, saving you time and money. And so how did I basically start with the idea? And that is, it was not basically the idea of doing. Yes?
Sorry, are you sharing the screen? Are you
presenting? I forgot.
Because
yes.
you didn't share the screen yet.
I forgot to share the screen. Thank you.
Yeah, now
yes, I just show this first slide again. This is what I said before. You didn't miss much. But now, so how did I basically then came up with this idea? This was not an idea of doing a startup, but it started when I did my master thesis. I had the desire to be independent. I did a bachelor thesis, a semester project, and during the project, at some point, I always thought like somehow I would have done it differently. So that's why I thought for the master thesis, I want to create and design my own project. So what I did is I went to a professor at ETH, where I knew that the group allows master's thesis students to develop their own project, but they need to basically pitch it at the beginning and propose it. And then the professor will decide if one can continue. And when I started, I went to a postdoc, and I asked him, yes, I mean, does he if he has some idea or material where I could start. And he told me, yes, you know, we have this material which is called barium titanate. And what is interesting about it is it's piezoelectric. So it means when you apply a mechanical force or a stress on the material, it generates electrical charges. And then when he said we can produce it with this process, and this is called flame spray pyrolysis. So it's a combustion process where you basically feed a precursor where you have the elements dissolved in an organic solvent. So we have the barium and the titanium dissolved in an organic solvent, and then you basically feed it through a nozzle and you produce a spray. And then you ignite this spray with a flame. And then you can enclose it with a tube, and you can play a bit with the tube length. So what you do is you make the flame bigger or shorter, or you increase the residence time at high temperatures. And this way they could produce nanoparticles, and then they could produce barium titanate, but they had no application for it. And then I thought, okay, I checked the literature, what could we do with barium titanate? And then what caught my interest was bone tissue engineering. So I found some papers where they used barium titanate, the ceramic, and they implanted it into the jaw of a dog, and they showed that there was mineralization on one surface but not on the other, and they related it to the piezoelectric properties. And so this is basically how I wanted to do bone tissue engineering, and then so it was not an idea of basically already doing a startup, but it was more the desire of doing something independently. And then I proposed this to the postdoc, and well, his first answer was no, because he thought within that group it would be too difficult because they had no expertise in bone tissue engineering. So, but what I want to elaborate a little bit more is the motivation that I had for bone tissue engineering. And one of the things is I love outdoor sports. So this is in the Swiss mountains. While I'm the only Swiss guy, so we have postdocs or PhD students, former ones from South Africa and from the United States. And my friend Eric, well, he broke then on one ski trip his collarbone, and this is his X-ray picture. And then I thought, okay, bone fractures, I mean, already it's an issue, right? So I read that there were 9 million fractures each year, and then the problem is that around 10 to 13%, they result in delayed healing or non-union fractures. So they don't heal by itself. Everyone that once broke a bone that is not severe knows that it just heals. And these delayed and non-union fractures, they result in high additional healthcare costs. And if you look at the market, you don't really find a big innovation. It's incremental improvements to some extent. And what is the reason is it's basically the complexity of bone. So if one looks at bone, at a big bone or a long bone, you see that in the center it's porous, and at the edge you have the cortical bone, which is very dense and compact and strong. And then if you look at the cortical bone, it's actually a composite. So it's a composite of hydroxyapatite, nanocrystals, and collagen. And the collagen makes the bone flexible, and then the hydroxyapatite makes it compact and tough. And then if you have a small fracture or crack, it heals, right? But then if you have a critical size defect, it doesn't heal by itself anymore. And then what I found in literature is that researchers do polymer nanocomposite bone scaffolds as an implant, basically, to help the recovery of the bone. And what they add is then, because bone has hydroxyapatite, they also add basically hydroxyapatite nanoparticles. And they use a bioresorbable polymer, for example, Resomer from Evonik. And then what I thought is, or what I found, right, is that bone is also piezoelectric. That's why it caught my interest. So the collagen, if you apply force on it, it also generates electrical charges. And that's why there were already people, they tried doing a composite with barium titanate. But I also started doing that, but I quickly realized that it's not so easy to grow bone in vitro. And basically why there is not a lot of innovation is because the development time and optimization time is very long. And So in my old group, then they thought, okay, this is too difficult. As a master thesis, we don't do it. So I took a small detour through nanocomposites because I needed the master thesis, right? So here I don't go much into detail, but I worked also on piezoelectricity. So I produced a nanocomposite membrane, and then I could basically then apply mechanical loading. So I mounted it here on a device where I could apply fluid pressure, and then this would bend up and down, and then I could measure the voltage. And then what we could show is basically that if we tune the properties of the nanoparticles, so we change their size, we can basically improve the voltage output. So we could produce nanocomposites that have a higher voltage output than if you would use nanoparticles from commercial suppliers, for example, Sigma Aldrich. And then this we then published, and my professor was happy. And then I completed my master's thesis, and I could start my PhD, right? And then in my PhD, of course, I wanted to go back to what really interested me, and this was bone tissue engineering. And so I started a collaboration basically with the laboratory for bone biomechanics. This is where I met Pedro. So if you have an idea, basically you want to do something, you need to have the right resources, right? So in my old lab, I would not have been able to do it. So that's why with an ETH, what is nice is it's very big, and we have many different groups, and they're always willing to help. So then the next thing that I want to convey is basically, well, if you have an idea, you need to test it, right? Because the easiest person that you can fool, that's yourself. So that's why I started them doing basically these tests with barium titanate. And what I did is in the beginning, I tried this static cell culture because that's what everyone is doing, what everyone can do. So you use a well plate, you add the scaffolds, and then you add with the cells with a pipette from the top. And then what I did is I changed the composition of the composite. So I changed the volume ratio between the barium and titanate and the hydroxyapatite. So I went from a little bit of barium titanate and a lot of hydroxyapatite to a lot of barium titanate and a little bit of hydroxyapatite. And here you see just cross sections, microscopy images. So what you see here, these dark structures is a scaffold, and then I observed these big clusters. And then I could confirm with another technique that this is indeed mineral. And then we also did staining, so I could show that I have cells inside. So this is indeed cell-made mineral in vitro. And then I thought, okay, this 1-1 composition is good, but I also tested a bit more compositions. But then the problem is these are images. As an engineer, I also always want to quantify something and test many different samples and make an average to compare it. And for this, I need basically to develop better technique. So when I joined then the laboratory for bone biomechanics, what my professor, who is now my professor, Alves, said is I should develop a framework and not just the material, because with a framework I can test different materials. And what we then have basically is what they already had in the lab is a bioreactor. So inside this bioreactor I can place 2 scaffolds, and one scaffold would be then loaded through this piston, and one scaffold is then static. And then I can record the displacement of the piston, and I can record the force that apply on the scaffold to really make sure that it is loaded. And I load then the scaffold 3 times per week, so Monday, Wednesday, and Friday, to basically emulate physiological loading. Because if, as a human, you walk every day, so you apply some load to the scaffold. And then what you can then measure is basically, if you have it from multiple time points, that's why we use bioreactors, we take micro-CT images, we then get these images. And if you look before the culture, this is how the scaffold looks like with a 1-1 ratio. And then after the culture, what I then also observed, like I did in the microscopy images, I could then detect basically these mineral clusters inside the pores. And then we quantified it. So what you see here is I call it scaffold mineral density maturation. So just how much more contrast I get over time. And I have, of course, this is then the baseline that I use to normalize it. And then what I observed is that the more barium titanate I add, the less mineralization I get. And then because it's piezoelectric, of course, we can apply this dynamic loading. So I started actually with the 9-1 at the very beginning of my PhD, because I found some literature. They said, if you have a lot of barium titanate, you have this piezoelectric effect. And then when I applied the dynamic loading, it didn't work at all. And this basically, I spent on these ratios 2.5 years of my PhDs, I think almost 3 years. And that was also what basically troubled a bit my old professor, because he didn't see the progress in my project. And what was also the reason why I then switched during my PhD to the group where I am now. But then, of course, if you don't give up, if you have a problem that no one has solved so far, it's difficult, but you should just not give up too early, right? So I continued then, so I go down this and reduce it. And then I basically saw that, yes, with dynamic loading, I have a significant increase in the mineralization rate in these scaffolds. But then this is also something that that was mentioned by Morten, is that you need to validate it also a bit to the benchmark. Because if I saw the literature papers, they sometimes compare the new material to Teflon. And then they always show it's better than Teflon. But I mean, basically everything works better than Teflon if you want to have bone growth. So what I use then is just a pure hydroxyapatite. And then I need to also make sure that I validate my benchmark, right? So these are now micro CT images where I have from one time point. So the first time point I have an image, and then I get after 6 weeks another image with a micro CT, and then I can put them on top of each other. And then I see the contrasts. And then where I see orange, I have basically new mineral that was formed, and in gray, that's the scaffold material. And then I saw already that under dynamic loading, so this is the red square, I saw that in the top region, it seemed like I have more mineral formation. And then I can quantify this. So I analyze basically these regions of interest in the top and bottom for the 2 different modes. And then I observed that in the top region, yes, indeed, I have a significantly higher bone formation rate. And then if we look then at histology images, we could also see that we have in the top region more cells. And this is, let's say, a disadvantage of the technique of just pipetting the cells on the top of the scaffold, because then they stay most on the top. But in my case, it was also good, because it really showed that what I have in the CT, it also is basically what we observe in the scaffold if we do histology. And then I did then basically also more experiments to really compare my new material to the benchmark. So the best material that I had so far is 3:7, so barium titanate to hydroxyapatite ratio. And then I did again a study where I changed the culture conditions. So this is also nice. If we do this, if we have these bioreactors, we can change the experiment while executing it. So I had for 4 weeks, I had it only in control medium, so without supplements that would induce osteogenesis in vitro, because I thought maybe it is enough with the piezoelectric properties that they will, make the cells differentiate. But I mean, I also discussed this with Pedro, and he was laughing, because he said, like, there will be no mineral if you don't add these osteogenic supplements, because you need to add the calcium and the phosphate. But as an engineer, of course, you don't know these things. So that's why I still tried. But what was good then, after 4 weeks, I always looked at my data and I realized nothing is happening. So everything stayed here low. And then when I started, when I added then the osteogenic supplements to the culture medium, I really saw the biggest effect, basically mineralization on the piezoelectric scaffold under dynamic loading. So the dynamic loading really makes a difference. And even more so when you have a piezoelectric material. And then I basically then completed my PhD this year. So I was in 2 different groups. I was in the group of Professor Francine for nanoparticle synthesis and material characterization, where I learned a lot about how to design a project, and especially also what Morten mentioned in his talk is that why do you do a project? So the first question I always had to answer is, why does it matter? Whatever I wanted to do, and then to basically go and execute this project that I wanted to do, I switched into the group of Professor Müller in the Laboratory for Bone Biomechanics, where I also met Pedro, and this is then what I did. And then at the end of my PhD, I thought, like, So what do I do now? Because I mean, then of course in Switzerland, either you join a big corporate company, a pharmaceutical, for example, Roche, or you can do a startup. And then I always fancied doing a startup, but I was then not sure if I should do one, if I have enough, if I have a good idea. So I started applying to other startups, and then I talked a little bit, and then at some point I thought, like, no, I mean, I've spent now 5 years for my PhD, and I wanted to do my own project, so I tried now myself. And then I went to Ralph, so he's my professor, and I told him, I want to continue as a postdoc and do my startup, and I think we know now we have this good material, and I want to somehow turn this material into a bone implant. And then his answer was, no, I don't support you on this. And then I was a bit like disappointed, of course. And then he explained to me, he doesn't think that with just this material, if I want to do a startup, he thinks it's too difficult to get into clinics because he already is so long in the field. So he proposed to me to change a bit the direction of my idea and use more the technology that I developed. But then you should test your ideas, but if you hear something from a professor or someone else with a recommendation, you should also test what they were saying. So I contacted then a friend that I knew. He was working for a big dental implant company in Switzerland, and he was global product manager. And I asked him, so how much does it cost to launch a new product? And then he said, like, it's 40 million. And then he said, it's not a big innovation, because they don't need to. And then I realized that maybe my professor is right, and going with a biomaterial is difficult. And I mean, maybe I need to take another route to do a startup. So I also then came to the conclusion that, to me, for example, bone fractures, I asked myself, is it a grand challenge? And I thought, okay, so the collarbone of my friend Eric, it healed. I also had just recently some rib fractures from a bike accident, and also they healed. So yes, some people, they need scaffolds, they need better implants, but there are products out there that work, right? So then I thought then about to basically make another product or another idea, which is then Compagos. And before I now go into Compagos to explain a little bit what we do at the moment, I just wanted to give you a bit an overview of the startup landscape in Switzerland, and where it starts as if you're an ETH student, or you completed your master thesis or your PhD, you can basically apply for an ETH Pioneer Fellowship. And the ETH Pioneer Fellowship is specifically meant to translate your research project into a startup. They provide basically for 12 months if you're alone, or for 18 months they provide this much money, and it's a competitive application process. And then what Switzerland also has is iNANO-Swiss. iNANO-Swiss is a Swiss innovation agency, so it's also funded by tax money, and they have different funding schemes. So they have you can get a voucher for preliminary studies for 15K, which is the 2-page proposal. Then they have innovation projects, for example, bridge, proof of concept, or a bigger bridge grant, which are also very competitive with, I think the acceptance rate is 3 to 6%. They offer startup coaching where you can apply, and then you get basically a voucher where you get a coach from industry, and you can pick the coach that you want so you can say you want 10 hours with this coach and then another 20 hours with another coach. And then they have a lot of startup trainings where you go through the idea generation, concept, business creation, and then growth. And then there are different foundations that fund organizations such as VentureKick or InnoBooster. And then in total, what you have available in Switzerland, when I did my math correctly, is you can get access to around 3 to 5 million Swiss francs of non-dilutive funding. So this means you don't need to, you don't have to give away shares of your company. So when you create a limited company, you create shares, and you have basically this much access to funding, which I think is quite nice in Switzerland. And then one thing that what I realized, so I started taking now these courses. So since I completed my PhD, I'm in the postdoc, I take courses for startups to get all these trainings. And then what I realized is you need to switch a bit your mindset. So I was basically a researcher and an engineer. And then you always think about technology. So when you, which 3D printer do I need for my projects? And what are the features of the 3D printer? You know, how much can I heat it up? How fast can I print? So and then also when I did my projects and I design, it's a lot of always focus is on inventing something new. And then in these courses, what they teach you and what I learned is that you don't focus anymore so much on inventing something, but it's more about what is the unique value proposal that you have. Why should people or potential customer be interested in buying your product? And what is your magic sauce? So what do you do different than other startups or what the companies already do? So there is more focus on the execution and creating value from innovations that you did during your time at the university or inventions that other people did, right? You don't have to come up with the idea. You can also just be good at executing ideas. And then you should always be ready basically to pivot from your original idea. So I started with, I wanted to do a biomaterial, and then with my professor, we went towards we want to provide personalized medicine to treat patients with our technology. And then I started testing it, and then I realized people don't understand it. So you need to test your idea again and again. And the easiest way I think is, so since I have an idea, if I meet someone new or friends or I go for dinner, I always tell them about my idea, and then I check do they understand it, what questions do they have, and then I always change a little bit how I pitch it. And at the moment, so the startup is called Compagos, and this is another way. So the technology that we want to offer is basically 3D printed lab-grown alternatives to animal experiments. And the problem that we want to solve is basically we want to accelerate the development of new drugs or implants, which is a very time-consuming and cost-intensive process. So if you look, for example, at drug development, I mean, only a number that I found online is that 5 in 5,000 products, they go into human testing. So you go to phase 1, phase 2, phase 3 testing. And the whole process from research and development, so from the discovery to launching the product, can last 12 years and a number that I found, for example, costs 2.6 billion. And 2 to 3 years, what I found is then spent basically on preclinical testing. So from research development, you do preclinical testing, in vivo testing in mice we saw, or in rabbits, or for large implants in sheep. And then you go into phase 1 study. And we want to basically reduce the number of animals needed by providing our technology to other companies or biotech companies or researchers as well. And the solution that we want to offer is basically scalable and robust and easy to operate framework. So we combine different technologies. We use stem cells from humans. We even have a research project where we use stem cells from patients from a children's university hospital. Then we create a bioink, so we mix them with a hydrogel, which is also based from another PhD student, this technology that completed his PhD in our lab. And we do everything in vitro. Then we 3D bioprint basically templates for bone, and then we use these bioreactors that I showed to you to apply mechanical loading to basically then produce bone in vitro. And doing this, we want to replace basically animal models, or for sure reduce them, and also basically provide early efficacy reports. So we complete our projects within a master thesis, within 6 months, we produce a lot of data. And then I go a bit faster. So this is now what they teach you in the startup courses. You need to show some market potential. And then, for example, this is now this 2.6 million, the cost for the new drug, for example, for osteoporosis. And then if you break it down to Switzerland, maybe it will come to 430 million, which is then the serviceable market. And then the market that we could maybe reach as a startup, what I hope is by 2025 that we can start executing projects, for example, for a clinical research organization, to start having some income. And then they always want to see the competition. And then what I do is I make a chart where I have basically the value that the company provides during the development of a new drug or implant and the speed. And what we want to excel is at the speed. So there is a startup at EPFL, for example, Sun Biosciences, Then Roche has a new institute for translational bioengineering where they hired a former professor from EPFL. In Netherlands, they have a big institute. They work a lot with organoids, but not for bone. And then you have these clinical research organizations. They execute these clinical phase studies for big companies. And what we at the moment where I am is we try really to see where do we fit into the existing market. So that's why at the moment what I always think we have the biggest chance is for our technology that we fit between the discovery of new implants or drugs and the early testing in animals. And they want to see some roadmap. So where do we want to go in the next 5 years? So at the moment we do the scale up. We have only one such a mechanical device to do the projects. So we can maybe host 2 master thesis students or 2 projects we can execute at the same time. And until next year, I want to do a scale up 4 to 5 times so that we can start maybe doing some pilot studies with potential customers, that we just have more throughput of projects. And then if we get this, so in 2025, I want to leave ETH, and then we want to go more towards personalized medicine so that we use cells from patients that need maybe a new treatment. And then maybe we want to include then in a later stage, sell the whole setup as a product so other people can start to just have a better scalability. And then at the moment, the team that I have, it's me. I applied now with a former bachelor student for this ETH Pioneer Fellowship. I got in contact with a doctor. He's orthopedic who left now clinics, and he's also helping me a bit as a part of his Master of Advanced Studies that he does. And then I have scientific advisors that I mentioned here, which is my Professor Alves. And we have another clinician that works at the Children's University Hospital. And also Pedro, he taught me a lot of bone biology. I think no one taught me so much about bone biology as Pedro in a very short time. And it was very comprehensible. So I'm very thankful for that. And he has also very good ideas actually for good projects. And then so what is the next 2 years that I want to achieve? And this is also how I want to close my talk. So what is actually, when you talk to startup founders, they also in Switzerland, what they say that the biggest challenge is basically your team. You need to form somehow a winning team because you can have a good idea, but many people have good ideas. You can have money, and in Switzerland, money is available. And then also in other countries, if you have a good ideas and you have a good, basically, education, you can get the money. But alone you won't do a startup, right? And with 2 people, 3 people is also difficult. But you need to have a team, and this is where I am. I have a WhatsApp group where I have interested friends and other people that I contact. So around 14 to 16 people over LinkedIn. So I'm growing always my network. To basically just see where do I get complementary skills. So people that think a bit different than me, have a different background. And I always get the comment, I need more business background in my team. And then what we also need to do is we need to validate the business model and the idea. So we have basically a prototype for the device. We can execute projects for research. But now what we need to do is to validate also the technology. So we have to see if what we do in vitro, when we, for example, want to test 2 or 3 different compounds for bone formation, or if you want to test a bone implant that has a new surface functionalization, a coating, that when we do the experiments in vitro and they are done in vivo, in the end, it's always a decision. Is it significantly better or not? And then what we need to make sure is that we come to the same, basically, decision as if you would execute basically the experiments in animals. And then, of course, we have to establish this running service platform and just do these pilot projects with industry or also with other researchers, actually, that maybe don't have access to in vivo studies. Or what is also a problem is that the ethics approval for in vivo studies is getting more difficult to get it, basically, in Europe, what I heard so far. And then also basically secure these grants that I mentioned at the beginning. So I apply to every grant that I see. If there is an opportunity, I always send my documents because it's very competitive. The acceptance rate is very low. So one needs basically to apply to what is available and then just improve all the time. And yes, so also if some of you guys are interested to hear more or have a discussion with me or have a Zoom call, feel free. We have a homepage that I need to update soon, or you also reach me basically on this, with this email. And yes, with this, I'm done with my talk.
Thank you very much, Gian, for a very nice presentation. Now we have a question and answer section. The audience, they can send the questions through the YouTube channel. They can ask the questions, and we can also interact and ask questions for each other. Can you please put Morten also on the screen? I think it's time now to talk. So thank you both for the presentations. We already have one question from Victor. He's asking, what are the challenges to do research like from the bench to the market? What are the main challenges you think? Because we are scientists, usually, like many people that are watching here, they are from the academia. So it's very difficult to talk to the companies and also to have access to this completely different world. So I think this is the main question. What are the challenges in the scientific point of view and also in the dealing with the companies?
Yeah, maybe I could start. I mean, at least one challenge, as I see it, is that as a scientist, you have to keep on publishing as well. So you could say in the beginning, it was easy in the way that you're developing something new, you can publish, but then when you have to validate later on, maybe in vivo models, right, where maybe there's not that much new science, but heavy expenses, then I think there's typically, at least in health science, there's a gap between when you finish your research and until you have a product on the market. I think that's a difficult one if you don't have a company that can help you at that point, because you can't apply for new funding, right, on the same project, because they would say that's old news. We know that.
Yeah, and I was talking also to Gian when I was in Zürich, that he said, I like to be in academia to publish, but it's not my real goal to publish anymore. Like now I will go for the startup, and it's different. It's a little bit difficult to do both, to do high quality research and also.
So it's also what I mean. I also in my old group, the group leader, they made now a startup, and I asked him a bit about his experiences, and he also, I mean, what Morten said is, if you're in academia, you need to publish. Mm-hmm. And then what I also learned in some of these startup courses is they say publications are nice, but they're also a bit. I mean, of course, if you have a patent, you need to first file the patent and then you can publish. But the publication process needs a lot of energy, right? Everyone who's in his PhDs, they know how tough it is, and then you get the reviewer feedback, and it's a long process. And what some people said is, if you're in a startup and you still want to publish some of your work, or maybe you don't know if you want to do startup or you want to stay in academia, then you invest a lot of time into the publishing, and then in industry or the investors, they are not so, for them the publication is not let's say a track record, it's not a must. What they like to see, what they said is, if you have high impact publications, if you go into Nature Communications, Nature Biomedical Engineering, and you generate publicity with it, that is nice, because then you have some outreach, and then also companies, they start reading about this in news feeds. But that is right, that's another risk to get such good publications. And then what I personally think what is difficult is if now what I experienced is, if you want to explain what you actually have, because when you're a researcher, you know how good your material is, or you know what you do, you know every little detail of it. And for you, it's clear. This is what we have. It's perfect. This works, right? We can do. This can change the world. This is somehow what the motivation that I also always had to do something. But then, if you want to talk if I talk them to pitch my basic ideas sometimes to investors, this is this mindset that needs to change. It's more like, yeah, but they're not interested into the technological details. they want to just know what value can you create for the company. And that's for me still a bit difficult because I don't see into the companies how they work. So I also, one thing I spend a lot of time is I still need to invest time is getting in contact with they call them key opinion leaders. So people that are in industry or are in hospitals and where you address their needs and to basically find the match between the need or find the unmet need and the technology or know-how that was developed. I think that's really critical and difficult.
Yeah, in this regard, like Morten, you have a product that is almost being released now by a company, and you told that the company supported you from the very beginning, right?
Yes.
And sometimes we have an idea, but we don't know when to talk to the company.
No.
Can you give any advice on that? Like, if you have an idea, when to talk to the company, how difficult it is, for example, to do a patent and how costly it is? And
yes, in general terms, I would advise to ask very early in the process. In general, I know that this could be an issue with the IPR, but in general, as Gian also said, I mean, you have to know the real problems, because what might happen is that you as a researcher sit and develop something and you think it's really, really good, and then you find out that it was a nice solution, but it was not the problem that the company focused on. I mean, I have several times had something that I thought, okay, this must be good, and then they say, yeah, but I mean, we are much more interested in this and this, for example. So I think the sooner you know what their actual needs are, the better. Of course, there is always a risk, but I would also say in general, I don't think if you can ask in more general terms, avoiding telling them the key invention in what you are developing, then I think you should go very early. Of course, being careful what you convey to them, but still, it could help you a lot. So it's definitely a problem working with something several years, developing something, and then you find out that you solved a nice problem, but that was not what the company was interested in.
yeah, I see. And regarding the for example, your product is directly applied to health. And regarding the regulations, for example, the regulations for using in human beings, and also in the research, because you showed data from phase 1, in vitro and in vitro tests, and also animal tests. And how is it to go for the clinical trials in Denmark?
That's really challenging, and that's definitely something I don't want to do myself, because I don't know the rules, and that's also why you need someone who has expertise in bringing that the next step, because I can go to, I would say personally, my collaborators are doing the in vivo. I'm only going until in vitro, because that's my competences, right? And if I were to file all the comply with all these regulations, I mean, I don't think that's possible for a researcher, at least not with my background. So I have to have collaborators that can do that.
And regarding that, Gian, you that is launching a startup, you said that you want to build a board of a team. And when you talk about a team for a startup, it's different from a team for developing an idea in the university. So which kind of people do you need to develop this team, and how you can get funding to pay these people in Switzerland? How is it?
Yeah, so what I mean from the courses that I had is what they, I mean, either the people they invest in the idea, if they think this idea and technology is excellent and it's really unique, then they invest in that, and then you need to have a good team where they think it can execute it. Or they think you have a brilliant team with people, you have a really good idea, but the technology is maybe a bit difficult for them to assess, then you need to somehow generate, you need to win some grants, right? So to get some these early startup grants, or fellowship grants, because then your technology is still assessed by professors and other people, and they trust them basically. When you got the ETH Pioneer Fellowship, or if you got an early grant for startups, so you won basically yes, and then they trust basically more that the technology was validated or at least assessed by other experts, and then they would just want to see that you have a strong team. And when I present my team, and then the people that assess it, they have them maybe, I mean, some have a background maybe in medical devices over many years, and some are maybe pure business people. They then always tell me, yes, you have a very strong focus on technology with your people, or everyone has a PhD or is engineer or is a professor, and then they always say you lack a bit the business aspect. And for example, in this pitch, I was applying with these slides that you saw, the last one for this pitching event, for winning this 10 to get into the pitching event for 10K. And then what he said is like, yeah, he really liked it, but some of the business model is still missing, and they want to see a financial plan. So like a 5-year financial plan where you come up with numbers that everyone knows they are not correct, but most of the people, they can assess if it's consistent, if you did your homework, and they want to see this. And then they will schedule me in front of the jury. So yeah, so I need basically not just experts on bone tissue engineering, bone biology, or bioreactors or imaging, but also some people that have more a commercial or business background. That yes.
Yeah. Sorry, Morten, do you want to say?
I could have one more. In fact, I was thinking that one good thing in Denmark is that we have the Novo Nordisk Foundation, and they are in fact handing out a kind of stipends for people at the university who wants to maybe work on a business idea for half a year or 1 year. So that would typically be around 70,000 euro or 140,000 euro. And they don't want any part of the company or anything. So you apply as a standard application, academic application, and then if they find that it's a good business model, then in fact they give you feedback how you could even improve it, or offer sometimes to have a conversation with you how to improve it, and then you get these funding. It has to be controlled by a university. You don't get them privately, but it's for maturing a product, which I think is a really good way of doing it for companies.
Regarding the funding, I just stepped talking to the audience. This Novo Nordisk Foundation, there is this synergy program that we applied for. So it's also open like for collaborations between countries.
Yes.
And in the case of Switzerland, Gian, I just saw that there is an open call for iNANO-Swiss together with Embrapi, that is the Brazilian agency that is like iNANO-Swiss, and they have a deal. Like I think there was an open call last year, and they will open the call again.
Okay.
So
I will check it out.
A percentage. The company gives a percentage, and the iNANO-Swiss and Embrapi come together to fund. And what I noticed over the last few years is that for basic science, there is fewer and fewer opportunities for applications for funding in Brazil, and it's growing a lot for companies and for collaborations with industry. I would like to know from you if this is also happening in Switzerland and Denmark, or if they kept the money for basic sciences over the years, because we have a strong feeling that we have not, actually now we have nothing like for universities, like it was cut a lot, but we see some opportunities that we didn't see before for entrepreneurship. So
you could say, I don't think they have cut as such in Denmark. They have redistributed, which is not a good thing for me. For some of my colleagues it is, but not for me, because right now there's a lot of focus on green development and circular economy. So that means a lot of the funding in Denmark is now focused on, which is also of course a very, very severe problem. So how to solve like energy crisis, how to increase sustainability. So in that way, maybe the focus on health is a little less now. So you could say probably less funding for my research field, but not in general.
Yeah, as a postdoc, I don't see too well basically on this top level the decisions. But just from reading and observing, I mean, ETH, they always have, it's the ETH domain, right, that has basically money from the taxpayers, and then they distribute, and they always have these negotiations between EPFL and ETH, because there's one cake and then they slice the cake. But then within ETH, I think, I mean, every professor has his fixed basically budget that he has, and then they need to basically raise basically grants through other fundings. And that's, I mean, one thing that happened that in Switzerland, they lost basically the affiliation to the ERC, which is a big topic now in Switzerland for basic research funding. That, I mean, now Switzerland, they want to cover some of this money, but I know, for example, for startups, they also have through ERC, they have access to grants, and this is somehow not covered. But what I was surprised in Switzerland that I did really not know as a student or PhD student is basically for startups, the ecosystem and in life sciences and medical devices, there are a lot of trainings and funding available, and there even now there's some new venture capital companies that they started now educating also at ETH at university potential venture capital people who would invest in startups, and they basically really nurture this startup ecosystem thinking just to get more people that want to try it. Because one number I saw in a course that I took, entrepreneurial risks, they showed the statistics of ETH startups, and then 90% were more than 90% were successful or survived more than 5 years. And then they said if you compare this to the US, you only have like, I don't know, 30-40%. And I said, okay, the conclusion is, okay, everyone is great, or only the people that really know it's working dare it, and actually too few people try it, because then it will go down. Yeah, I think I talk to people at ETH, I hear more people that want to go for entrepreneurship than they want to go for an academic career.
Yeah, this is very encouraging for Compagos to know that 90% of our colleagues are successful.
Yeah.
So yeah, it gives strength. We have time for maybe one last question from the audience. But if not, like we're approaching the end of our talk. It was very nice talking to you too today. And I see you again in the future. So not in the future, but I see you in a couple of days, Morten and Gian. Thank you very much for the presentations and for the discussion.
No problem.
And thanks the audience and the organization again for allowing us to talk today and for listening to us this morning. Thank you.
Okay.
Okay.
Bye.
Thank you. Bye-bye.